Hey guys, thank you everyone for being here. So Lindsay is going to be talking about her research today. But before she gets into that, we're going to ask her a few questions about what it's like to be a scientist. At any point, if you guys think of any questions throughout this, feel free to just unmute yourself and shout it out and interrupt. Or if you don't want to do that, you can type it in the chat and we can monitor it and we can ask Lindsay. So ask any questions you think of. But Lindsay, I guess let's start with, have you always wanted to be a scientist and what got you into this field?
That's a good question. Yeah, I would say that I was always kind of interested in science. When I was younger, I used to like reading books on like animals and dinosaurs and stuff like that. And then I guess it wasn't until like high school where I realized I was interested in biology. And so I knew I wanted to do something along that realm. But I guess it wasn't until college where I decided I wanted to like do a PhD. And I think part of that decision was just the fact that as an undergrad, I was being exposed to like a little bit of research, and also I was studying like biofilms when I was an undergrad. So I think I had like more questions and I was just super interested in it. And so I knew that becoming a scientist and, doing a PhD would help me kind of like answer those questions. And, I've always been interested in like discovering new things. So I think it kind of went along with that.
Cool. I guess going on with that then, what do you think is the hardest part about scientific research overall?
Yeah, definitely, some things just like don't pan out, you can always, you do a procedure that you've done a million times, and then you do it once and it just doesn't work, and you can't figure out what's wrong. So there's definitely like a lot of like troubleshooting, and it can be kind of discouraging sometimes as a scientist when things aren't quite working out the way you expect them to be. But overall, that's always kind of like overshadowed by the times where like things do work and it is how you expect. So there's pros and cons.
Yeah, I guess that's the other hand of it. So like, is that what you think is the most rewarding part then? Like when things go the other way and they work?
Yeah, definitely. And just like the idea of like figuring like the whole realization that like you're working towards something that like no one has known before, so I think it's just like a really cool concept to me that we're working on something novel.
Yeah, cool. Thank you. Okay, well, I guess we can just let you get into it and explain what these beautiful images are that we're looking at. Okay, so I'll let you take over, and just to reiterate, if any of you guys have any questions, just shout them out. Lindsay does not mind, or put them in the chat and we can ask them. So cool. Thanks, Lindsay.
Okay, thank you. So yeah, hi everyone. My name is Lindsay and I'm a PhD student studying biology at Columbia University. And the story I'm going to tell you today is all about bacteria and the intricate structures they form called biofilms, which I have several examples of here. And while these structures are really elaborate and striking, they also benefit the bacteria in many ways and help the bacteria actually deal with many stresses. But before I get into that, I just wanted to give you a little more background into who I am and what my lab is like. So here's me. And I'm originally from New Jersey. And growing up, as I mentioned, I was always kind of interested in science, but it wasn't until college that I was introduced to microbiology and biofilms. And here's an example of a really tiny biofilm under a microscope that I studied in undergrad. And I was really interested in these biofilms and I had a lot of questions about them. So I decided to pursue that research further. And I decided to do a PhD, which, as I mentioned, is just really cool because I'm studying something and I'm becoming an expert in something that's novel and something no one else knows. So I'm currently at Columbia, and here's a picture of my lab, as well as a picture of my lab members. And, all of us are trying to answer these different questions that we have about biofilms, but we all help each other out a lot, and we are just like one big team. And so finally, here's a picture of my cats because I couldn't not include them. And we'll see if maybe one of them just runs across my keyboard today. All right, but let's get into it. So first off, let's talk about bacteria in general and what we know about them. So when you hear bacteria, you might think of several negative associations. How there are these like tiny microscopic organisms that can get you sick. And while that's true, bacteria are actually all around us, on virtually every surface, on your skin, hands, your cell phones, even inside of you and inside your digestive tract. And the reason you're not sick all the time is because the majority of bacteria don't actually make you sick or cause illness. In fact, a lot of them actually exist to help you and prevent infections from bad bacteria. And so bacteria are such a prevalent part of our lives that it's actually estimated that for every one cell that makes up you, there's 10 bacteria cells also present. And so because bacteria are all around us, it's really important that we study them. And for a really long time, scientists studying bacteria focused mainly on bacteria grown in liquid test tubes or flasks. And in this state, bacteria swim around and exist as single cells, not really associating with one another. More recently, however, scientists realized that the vast majority of the time, these single cell bacteria actually form really large clusters with each other and secrete a sticky substance that holds them together, which I've shown here in purple, so that they can grow in size. And this large cluster of cells is what's known as biofilm. And so more often than not, bacteria actually exist in this biofilm state. And it's these biofilms that are all around us. For example, biofilms are present in hot springs at Yellowstone Park, and they're actually what's causing all the different colors you see here. On the other hand, though, biofilms can also potentially cause all types of problems and can mess with industrial processes by forming on the inside of pipes. And you might have had to deal with biofilms firsthand, since biofilms are also formed on our teeth, otherwise known as dental plaque. And one bacteria that's really good at forming these biofilms is the organism Pseudomonas aeruginosa. And this is the organism that our lab mainly studies. It's also very clinically relevant, and the World Health Organization has listed it as one of its top priority pathogens. So it can form biofilms within burn wounds or on medical devices. And it's especially a problem in patients with cystic fibrosis. So cystic fibrosis patients actually have a mutation that results in the buildup of a thick mucus on the inside of their lungs. And this mucus is the perfect environment for P. aeruginosa to colonize and take over. So in the lab, we study Pseudomonas aeruginosa biofilms by taking a liquid culture of bacteria and pipetting it onto an agar plate containing nutrients. And the spot will grow on top of the agar over the course of several days. And the result is a biofilm. So here is a colony biofilm, and it's about the size of a quarter. And within it, there's estimated to be about 10 billion cells. And when talking about biofilms, it can be hard to imagine just how many cells fit into such a tiny area. So to put it into perspective, that's like taking the entire population of the Earth and sticking everyone onto the island of Manhattan. And so going along with this analogy, you can imagine that if the entire world population was on Manhattan, there'd be a lot of issues. So how do we ensure that everyone has access to oxygen to breathe or access to food to eat? And just like us, bacteria also need oxygen and food. And so they also care about these issues. So let's dive a little deeper into these issues by taking a closer look at what exactly is going on inside the colony biofilm. So if you can imagine that if we pile cells upon cells And if only cells at the top can get oxygen and only cells at the bottom can get nutrients, you can imagine that gradients form. And so to better appreciate these gradients, let's take a colony biofilm that's been growing on an agar plate and turn it to its side. And this way we can have the part where the air is at the top and where the agar is at the bottom. And so you can imagine that oxygen can come from the top and it depletes on the way down. And we can actually use probes like this one to directly measure the oxygen gradient as we go through the biofilm. So what we find is that oxygen indeed depletes as we go through the biofilm. And we also find that past around 70 microns deep, there's actually no oxygen present. And this means that biofilms past 70 microns are made up of both what we call an oxic zone and an anoxic zone. And anoxic is just a word we use to describe an area where there's no oxygen present at all.
Lindsay, could I ask a question?
Yeah.
I think this is kind of silly of me to ask, but like, how do you probe the oxygen? Like, is it literally almost like a needle-sized structure that you just stick into a colony and it measures it?
Yeah, I don't know the actual specifics of it, but yeah, it's essentially, it's a really thin, tiny glass tube thing. And it almost looks, yeah, almost like a Pasteur pipette type thing. And it's really thin and you just stick it into the colony and somehow mechanicalistically, it can actually measure the oxygen concentration as you go down.
Oh, that's so crazy. I think of them as being so thin. Like, it's crazy that you can see different layers with that.
Yeah, exactly. Yeah, it's a really sensitive machine.
Yeah, could I follow up with another question?
Yeah.
So all of the cells are alive? Are any of the cells at the very bottom just like dead because they can't get any oxygen, or are they like crushed under the weight of the rest of the biofilm?
Yeah, we think these cells are actually still alive. Yeah.
Okay, very cool.
Yeah. That's a good question though. Okay, and so so I just showed you, so the oxygen profile of a colony biofilm actually looks more like this, where it is completely depleted at one point. And similarly, nutrients come from the bottom, where the biofilm rests on the nutrient agar, and deplete on the way up. So our lab is looking to answer all types of questions about what is going on within the biofilm, especially with respect to these different gradients. And one major question that we have is, so how are cells actually surviving under the various conditions within the biofilm? Or in other words, like, what strategies are the cells doing in order to carry out the processes that they need to survive when they're in such an environment where they're constantly competing for oxygen and nutrients? And in order to address this question, I'm just going to introduce you really quick to a technique we use in lab called thin sectioning. And here we take a colony biofilm and embed it in wax. And then we can use an instrument to take very, very thin slices of the biofilm, similar to how you would slice a loaf of bread. And the slices are about 10 microns thick, which is about 7 times thinner than a human hair. And then we can look at these under a microscope to see what the biofilm looks like along those gradients I just mentioned. And so now that I've introduced you to a common method we use in lab, let's go back to our question. So cells within a P. aeruginosa biofilm have several strategies for staying alive within this state. And one method involves the production of compounds called phenyphines. Another involves a process called fermentation. And finally, a third strategy that I'll be talking about involves the concept of cross-feeding. So we'll get to all these terms and words in a minute, but let's talk about phenyphine production first. So looking at the liquid culture of P. aeruginosa that I grew up overnight, you can see this greenish bluish pigment, and this is attributed to phenyphines, which are molecules of different colors that P. aeruginosa produces. And so this here is a wild type culture of P. aeruginosa, which just means that it's our standard reference strain without any major mutations. So However, if we grow an overnight of a mutant that does not have the genes to make phenyphines, which we call delta Phes, it actually no longer produces these molecules and it completely loses its blue color. And so the purpose of these molecules are not just to produce nice colors though. For one, these phenyphines function as antibiotics against other bacteria. And additionally, phenyphines have another really interesting role where they can become oxidized by molecular oxygen, which basically allows it to act really similarly to oxygen. And it's this property of phenyphines that's really important in the biofilm context. So let's say we just did a thin sectioning experiment, and we're now comparing sections between wild type and the delta Phes mutant, which again cannot make any phenyphines. And so we notice that our wild type section is, let's say, around 120 microns deep, and we know from oxygen probing data that the oxygen can only penetrate around 70 microns. And so we have a zone where oxygen is present and a zone where oxygen is completely absent. And so in our model, we can imagine that bacteria at the top, have oxygen access and they're happy, but cells at the bottom do not have oxygen access. So what do these cells at the bottom do? So this is where phenyphines come in. And phenyphines can actually use oxygen present in their environment in order to become oxidized. And then they can diffuse down deeper into the biofilm. And these phenyphines can serve as an oxygen source for the cells here. Or as we like to call it, these phenyphines act as molecular snorkels for the bacteria to breathe at the bottom. Further, in the mutant that I mentioned before that cannot produce phenyphines, we actually notice that these biofilms don't grow past 70 microns. And so they can't even form a zone that doesn't contain any oxygen. So therefore, phenyphines are one way that Pseudomonas aeruginosa biofilms deal with oxygen depletion and bacterial overcrowding. Okay, so now I'm going to talk about my second strategy that Pseudomonas biofilms use, and that's a process called fermentation. So let's look again at our biofilm thin section with its opposing oxygen and nutrient gradients. And if you think about it, you realize that the cells at the bottom of the biofilm are in a really interesting situation. They have plenty of nutrients available from the nutrient-rich agar plates they are growing on, but no access to oxygen. And so the question is, can cells here do anything with all these nutrients in a more oxygen-independent manner? And so it turns out that glucose, which is a common food source that we give our biofilms, is converted into pyruvate within the cell. And depending on whether oxygen is available, this pyruvate can enter two different pathways. So if oxygen is present, the cell can perform respiration. And respiration is kind of just a fancy word for breathing. It's surviving by using and taking up oxygen. And if oxygen is absent, however, pyruvate can still undergo several reactions, an important one of which is a conversion to lactate. The conversion of pyruvate to lactate in the absence of oxygen is what's known as fermentation. And this is actually what happens in your cells when you're exercising and your muscles cramp up. So these muscles don't have access to much oxygen anymore, and the muscle ache you feel is actually the buildup of lactate. And the good thing is that the cells undergoing fermentation can still produce enough energy to survive. So now the question is, okay, so are the cells within the biofilm actually undergoing fermentation to survive? And so to test this, we can grow our biofilms on glucose and carry out thin sectioning like we normally would, but we can add in a twist. So remember that we know that fermentation is occurring if lactate is being produced. And so in this experiment, we can make it so that the cells that make up this biofilm are genetically manipulated to contain what we call a reporter. And so with this reporter, when lactate is absent, nothing happens to the cells. But when lactate is present, the cells actually glow bright green under a fluorescent light. And this allows us to visualize not only if lactate is being produced within the biofilm, but also where in the biofilm lactate is being produced. And as you can see here in the thin sectioning that we just did, lactate seems to be produced deep within the biofilm. And you can see this in the green color. And so this is exactly where fermentation is believed to occur. So by combining thin sectioning experiments with reporters such as this one, it's a really cool way to understand what's going on within the biofilm. Okay, and finally, I just want to talk about our third strategy, something called metabolic cross-feeding. Oh, I think there was a hand raised. I don't know.
I have a question. That was my
Hi.
hand. So the lactate that the bacteria uses from the nutrients, is that kind of like like you don't need oxygen if you have lactate? It kind of works for oxygen, like it substitutes it.
Kind of, yeah. So so usually if oxygen is present, pyruvate undergoes all types of reactions. But and that's how the cell survives in the presence of oxygen. But if oxygen isn't available, then instead of doing all those reactions, the cell would just make lactate. And it doesn't produce as much energy as the other reactions do, but it still allows the cell to survive. Does that answer your question? Okay, good. Okay. So yeah, so our final strategy is something called metabolic cross-feeding. So let's go back to our model where we were looking at a biofilm thin section, keeping in mind the oxygen and nutrient gradients. Another issue that could occur, so how do we actually ensure that the nutrient gradient extends to the top? And what happens if the cells at the bottom take up all of the nutrients before the cells at the top get their fair share? And one way around this is something called metabolic cross-feeding. So first, I want you to think about what it's like when you're assigned a group project at school. To complete it more efficiently, a good strategy is to divide up the work. And so a few people might gather the research, another person might write down all the findings, and another person still, might be the one to present the project. And just like how we divide up work when we work on a team, individual cells within the biofilm do something similar. So instead of a school project, this biofilm is tasked with undergoing basic metabolism. And as I mentioned previously, metabolism can involve several reactions. So glucose is made into pyruvate, and depending on whether or not oxygen is present, cells can either undergo respiration or fermentation. And just like we divide up work for a project, bacteria will also divide up some tasks here. So as we just learned from our lactate reporter experiment from earlier, cells at the bottom are likely undergoing fermentation. And so we'll color them in pink. And the cells undergoing fermentation are kind of stuck with lactate as an end product and can't really do anything with it besides just excrete it. And so, however, the other cells can actually finish where these fermenting cells left off. And because this lactate basically represents a new food source. And if lactate diffuses up to where oxygen is present, bacteria can use lactate for cell respiration as well. So thus, while some cells might be getting some of their energy from glucose, other cells are getting their energy from lactate. And this helps ensure that no single nutrient source runs out for the cells at the top. Thus, we have a biofilm where different cells are dividing up different metabolic tasks. And this idea of cross-feeding is a major part of my research. And I'm looking into cross-feeding with lactate as well as other metabolites. And I'm also looking into, how important cross-feeding is for the cells in the oxic zone. Okay, so to sum everything up, I really hope I convinced you all today how interesting and dynamic the biofilm environment truly is, and how important it is to understand this environment. So the questions my lab and I ask about biofilms are like fundamental in a way, but also very applicable. And as I've mentioned, these biofilms are all around us. They live on virtually every surface and are especially relevant in clinical settings, making up around 80% of all infections. And in order to develop antibiotics to treat these infections, we need to understand what the cells are doing within these biofilms, and which is why the research that my lab does is so important. And it's really important that we have scientists studying these microbes around us, since at the end of the day, it is a microbial world and we're just living in it. So thank you so much, everyone. I can answer any more questions you have now. Thanks.
That's awesome. Thank you so much, Lindsay.
Yeah, thanks,
That was
Lindsay.
great. Lindsay, can I ask a question?
Yeah.
These dynamics that you see with these biofilms and like everything about their metabolism, is this specific to Pseudomonas, or do all bacteria that form biofilms like have similar dynamics?
Yeah, that's a good question. We pretty much all bacteria have to deal with these dynamics, and I was focusing on Pseudomonas because that's what our lab studies. But people have also demonstrated like fermentation and cross-feeding and similar things going on with completely different species.
I think, Emma, do you have a question?
Yes, I have another question. So in the beginning of the presentation, you mentioned how bacteria was everywhere, like on the screen of a computer, inside your body, and everywhere you look. Is it the same for biofilms? Or is it only like where the specific nutrients are, like on the plate where you did the testing?
That's a great question. So yeah, it's actually, we think that most of the time bacteria actually exist as these biofilms instead of bacteria. And so biofilms can like range in size. So sometimes they're really tiny and you can't see them. The biofilms that we work with in the lab that you see are, about the size of a quarter, so you can see them. But yeah, we think that actually most of the time bacteria are less in the single cell state and more in the biofilm community state. Great question.
Makes me want to clean everything around me.
No, it's okay though.
And that picture at the very end is so beautiful. Could you talk about like maybe why they look like that?
Yeah, so in order to get it to look like this, we actually include some dyes within the plate. And these dyes actually stain the matrix that the bacteria produces. So basically it's staining all of the sticky substance. And we find that, so colonies actually start out like pretty smooth, so without all of these wrinkles, but eventually over time they get all of these like weird wrinkles. And we think that that's actually another strategy that that biofilms can use, and they're creating more surface area by wrinkling up, and that allows them to get more oxygen.
Very cool.
Yeah.
Cool. Looks like we have a question in the chat. So Lionel's asking, which skills that you learned during your PhD will you be able to apply in a future job? Also, what are you planning to do after graduation? And, oh, sorry, this is a multi-part question. And then finally, on a side note, what part of cross-feeding would be a good drug target to interrupt biofilm formation? Oh, that's a cool question.
Okay, so Lionel,
I just shot a lot at you. Sorry, Lindsay.
Yeah. What skills learned during your PhD will you be able to apply to your future job?
Yep. And what are you planning on doing, I guess, after you graduate?
Yeah, so I think there's a lot of skills that you learn during a PhD that can be applied. And so sometimes it's not always the direct skills that you learn. So it's not like the, learning how to do a biofilm thin section or pipetting that matters too much. But what matters is the kind of investigative skills I learned, the, how to go about solving a problem. And scientists with PhDs become really good at that type of problem solving. And so that's, almost no matter what type of job you get, that's super critical. What are you planning to do after your graduation? That's a good question. And I don't know for sure. I think going into some type of like industry where I can like apply my microbial knowledge would be super interesting. Or even just like on the clinical side of things, I think would be good. And then finally, so your question about cross-feeding. Yeah, that's a great question. And so a lot of antibiotics tend to target, actively growing and dividing cells. And what's interesting about cross-feeding is that it opens the door for mixed metabolisms. So cells, some cells are really being like super active and dividing, while others, doing like fermentation are less active. And so these drugs actually tend to target, the ones that are even more active as opposed to the ones that are less active. And so I think if we could, target drugs, if we can target cells that are undergoing like this fermentation, in addition to like respiration, that could be the best way in order to interrupt biofilm formation. It's a great question though. Thanks, Lionel.
Does anyone else have any other questions for Lindsay? Okay. Yeah, I guess if no one has any more questions, thank you, Lindsay. This talk was like very, very cool. And thank you all guys for asking great questions too.
Yeah, thank you guys so much.
Yeah, thanks for being here.
Yeah, thanks, Lindsay.
Yeah, no problem. All right.
Okay, well, if people have any questions, feel free to stick around and ask. If not, feel free to log off, and hopefully we'll see you guys in a few weeks at our next seminar. Bye, guys.
Thanks. Bye.